So I'm Gemma I Mtnez. I'm a PhD student at the Institute of Evolutionary Biology in the lab of Rosa Fernandez. And I will present today in this session of the seminars our tool FANTASIA, functional annotation based on embedding space similarity, which was created as a collaboration between my lab and the lab of Ana Rojas in the Centro Andaluz de Biología del Desarrollo, thanks to another connection from the SIC, the Live Hub. So during my talk, I have divided it in 4 parts. First, I will talk about the function paradigm and what's function and what do we mean like that, and why did we create this tool, sort of, and why it's important. Secondly, I will talk about the protein language models, which is like the core of the tool that I'm going to present. Then I will present the FANTASIA, the tool itself, and its 2 versions. And finally, I will talk about the benchmark we did and how it performs when we apply it to non-model organisms. So first of all, it's important in biology to know the function of proteins, of genes, because that allow us to go from the sequence itself to the biological question that we want to answer, either if it's to use to understand a disease, the evolution of a process, or anything else. Traditionally and classically before bioinformatics, and actually the best way to determine the function of a protein is doing an experiment. And that can be done in model organisms. However, this is time-consuming, very expensive. There is only a handful of model organisms. We cannot apply this to non-model organisms. And so for the rest of the proteins, like the majority of them, which is actually growing, like the number of sequences that exist in the public databases, it's growing. For example, in the left, you have the number of proteins in GenBank per year that is exponentially increasing, but also there have been recent new databases like Logan here on the right, that is quite recent, that has 2 petabases of data, including the assembled metagenomes of across different environments and across the whole tree of life. And this is also getting worse with the new projects, the sequencing projects of not only metagenomes, like the one before, but also genomes of different organisms, like the Earth Biogenome Project, that every year they are increasing the number of sequences that we have available, and we cannot do experiments for all of them. So how can we assign the function of these sequences without doing experiments, because because it's not feasible? So what we have to rely on is on this small set of proteins that are experimentally validated in model organisms. And what we do is to transfer these functions to other model organisms, for example, in which we did not do the experiment. For example, we do an experiment in Arabidopsis, but in a specific protein, but we assume that the same protein or a similar protein in Mercantia, another plant, do a similar thing. And for other organisms, the same, even if they are non-model. So for most of these sequences that come from very diverse organisms, we transfer these functions from proteins for which we know the functions. And this is based on the assumption that the homologous sequences do the similar things. So the first step to assign a function would be from our sequences to compare in a reference sequence database where we have the proteins with a known function to identify the homologous sequences, and we do it by searching for sequence similarity, so that the proteins that are more similar, we say that they are homologous. And then when we found like the best match, given some thresholds and so on, we've transferred the function. For doing that, yes, we are assuming that similar sequences produce similar protein structures and produce the same physiological function. We are also assuming generally that one sequence produces one structure and one function, but there are several limitations and exceptions for this. In the case of getting similar sequences, there are I would say 2 main limitations. The first one is when we assign the similarity, it can be that even though the similarity is quite high, such in this example, that could be an 80% within the query and the target, the function itself can be the motif or the part of the protein that's giving that sequence the function is the one that is not similar. So we could erroneously assign functions to things that do not do that real function. And the other problem is the limitation of how confident we are in assigning that these two sequences are homologous, because we are basing ourselves on sequence similarity. There are like what we call like maybe the twilight zone, that is basically like some when we align proteins, that if the alignment is too short, we need to have a pretty high number of identical residues, identical amino acids, to be confident that these two sequences are the same or are pretty similar. So in these cases, there are many sequences for which we cannot assess that they are similar. And in this case, we cannot transfer any function. And this happens quite a bit. It can be because there is no information on the databases. So it could be that if we had these sequences, we would get a match, but there is no information. Could also be that there is some genes that are specific of a species of a lineage, so the information is not there, or that because the evolution of the sequence has made it so diverse that we can no longer identify them as homologous. In the regarding similar structures, occurs the same thing, that two structures that are similar, that can be similar, can actually have different functions if there are only some small things that change in their active site, such as in this example, that one is an enzyme and the other is not an enzyme, but they are pretty close structurally. And finally, as I briefly said before, we are assuming that sequences that are homologous, that have the same evolutionary origin, have the same function. And that is and that relates to the ortholog conjecture, that says that genes that are the same gene from different species retain the function, but when these genes duplicate in a same species and there are now two paralogs, these two paralogs diverse, and one of them, both of them diversify. So that they say that orthologs have more similar function than paralogs. But this can be challenged in some examples and functional analogy. We have different proteins that have the same function, or there are some paralogs that indeed retain functions while the ortholog does not. And another example is that the function of one protein, depending on the other proteins that it binds, on how it binds to other proteins or other subunits of monomers, the function can change. And the same goes for the environment in which the protein is active. So that if there is a change, for example, in the copper, the copper, the histones have a copper reductase function instead of being the classical epigenetic proteins that bind to the DNA. So, even if we have all these limitations, this is still like one of the only ways that we have to assign functions to many proteins. And the functions that we use are mostly gene ontology terms. These gene ontology terms are like words terms, the control vocabularies that are linked together in an ontology, so that these terms are related to each other. So we have more general terms that have like children that are more specific, and these ones are related to each other. So we would assign these geoterms to our proteins to assign them a function. This gene ontology classifies these terms in like three different categories, three ontologies that are independent to each other. Cellular component, which talks about the location in the cells, like mitochondrial membrane. The molecular function, about the activity itself of the protein, like it's a water channel activity or a type of an enzyme. And finally, the biological process, in which general cellular or physiological process it participates: cellular detoxification, breathing, etc. Gene ontology terms that we have for some proteins, there are some of them that are not always manually curated. So there are some that are transferred based bioinformatically, but they have not been reviewed by a curator. And for example, in different model organisms, you can see in red the proportion of the annotated genes that have electronic annotations that have not been manually curated. And that also changes depending on the model organism. So for C. elegans below, for example, there are many more non-curated electronic annotations than in Drosophila, which is right above it, for an example. Also, for this kind of, when we talk about function, it's important to notice that the gene ontology changes through time. Imagine you assign a geoterm to a protein, but 3 years later, this geoterm might become obsolete and it's no longer there, it's not valid anymore. Not only that, but also the relationship between the different geoterms, the topology of the ontology itself can also change as well. And that might affect some of the inference that go later. Okay, so as I mentioned, we usually assign this function based on sequence similarity. However, there are many organisms for which, because of the things I mentioned before, there is a limitation, and we cannot assign homology to many of those genes. For example, in this tardigrade, which is a non-model organism, 40% of the genes do not have a hit to the reference databases, so we cannot assign any function. But even in model organisms, there is still a proportion of genes that we don't have a functional annotation. So for that, another alternative that we could explore would be the use of natural language, protein language models, which are based on natural language processing that I will explain a bit. Imagine we have these different words: cat, kitten, dog, and houses. So these models, what they do is they transform these words into a mathematical vector that is called embedding. So it's like number its position of the vector. They should include information like of the meaning of some property of the word. Like for example, in this example, the first column, like the first value of the embedding, would refer if the word refers to an animal or not. So that the three first that are animals have a higher value than a house, which is not an animal. So how does these models create these vectors? So basically, like the way they do it is they are trained on text, on paragraphs, or longer words or longer texts, and the idea is that they will try based on the text to identify, like to encode in this vector different properties of the text. Like for example, in this sentence, maybe these different like positions of the vectors can include information on the local properties, such as if this part is a subject in a sentence or it's a verb or not, or more global properties of meaning of the language or whatever. One example of this kind of models are transformers. So transformers, their objective could be like one of they would be useful for identifying, like in this example, what could it be? So imagine we have these two sentences: the cat sat on the mat because it was tired, and the cat sat on the mat because it was cozy. Here, it means different. In the first one, it is the cat, and the second one, it is the mat. So these transformers, this kind of models, what their objective is to identify what it is based on the context. Because transformers and artificial intelligence models that are designed to understand the language using the context, like the words in the surroundings, using a mechanism called attention that allows to look at all the words at the same time, and at the same time to decide which words are more important for understanding the meaning. So I'm going to put a very, very, very simple explanation of how do they work. So usually these models, one of the first steps that they can do is to mask some words. In this case, we have cat, mat, or it. And the objective of the model, when it is being trained, is to then predict accurately the words that have been masked. The architecture of these models, it can be quite diverse. In the case of the ones that use a transformer, they can have like an encoder, which is like a part of the architecture that is formed by, has different transformer layers, or a decoder that also has transformer layers, but they are like structured differently. Some models can have only encoders, others have only decoders, others have both of them, and they work better or worse for different objectives. So once we have those embeddings, the idea is that if we represented these vectors, they could show that the words that are similar in meaning would be closer in the vectorial space, in the embedding space. So now the idea is to transform this, to use this transferring to proteins. So we would have our protein sequences, our protein of interest, and a reference, and we would use a model to convert them to embeddings. So these protein language models would do the same as I mentioned before. Based on the sequence of the proteins, they will try to extract biological information from them. Either more specific things like here is a binding site, or here is a disulfide bond, or more global like thermostability, for example, the cellular location. There are out there many pre-trained protein language models, and I'm sure more are being created. And I will explain briefly, put some characteristics of a couple of them. First, the ESM, the evolutionary scale modeling. There are like several versions. I think now they are with a third version, and it has differences in the number of parameters. Has been trained mostly on UniRef, which are non-redundant protein sequences, and the architecture is based only on an encoder. The other kind of model would be Prot5, which has been trained on a larger database of 2.1 billion sequences called the Big Fantastic Database. And it was also fine-tuned on UniRef, and as architecture is a combination of an encoder or decoder, what it's called a T5. That's why it's called Prot5. So once these models create the embeddings, the embeddings would be different in size, in numbers, depending on the model you use. Now we would be able to compare these embeddings in the vectorial and embedding space and see which are the reference that is the closest, or the references, if we decide to use one or more, that are the closest to our protein of interest, and transfer these geoterms to our protein of interest. So this idea did not came from us, but it was already implemented in a software called GOPredSim. But this software has like several problems. The first one is that as of today, the several dependencies that it has are no longer maintained. So it is really hard to install it through a terminal, like in the command line. The authors have a web server, the developers, that you can put a sequence and get your embeddings or the properties. However, it's not feasible to do it if you want to run it in thousands of sequences, such as it was our objective to run it in in millions of proteins we wanted to run it. So for that, we created FANTASIA. FANTASIA comes from functional annotation based on embedding space similarity. Is a pipeline that re-implements this algorithm of GOPredSim, and given a proteome sequence file, it's like automatically can do the pre-processing and all the steps and give you a Gene Ontology annotation. It has the three main steps that I mentioned before. First, it generates the embeddings based on the model that it has. Secondly, it calculates the embedding similarity using Euclidean on-cosine distance. And finally, once it finds the closest one, or the closest, depending on the parameters you choose, the closest ones, transfer the gene from the reference, it transfer the Gene Ontology annotation. So I'm going to explain a bit the differences between FANTASIA version 1 and version 2, because at the time as of today, we have two versions of it. In the first version, the input it needed, it was pre-processed without option to remove it. It removed sequences that were 100% identical with CD hit, and sequences longer than 5,000 amino acids. This is a constraint of the way it was implemented in the first time. In the second version, this pre-processing is like optional. And in fact, the removal of sequence similars, it's only recommended for benchmarking. And in this case, would be only for benchmarking, it would be interesting to remove the sequences that are similar, but not inside the input file, but in the lookup table, like in the reference table. So that we would remove the sequences from the reference table that are similar to the ones in our input. Otherwise, we would not. This way, we can use it for benchmarking and try to see how the model with the different parameters and etc. works to predict the real function of our protein when it's not in the reference. The main difference, like in the way it's implemented between the 1 and 2, besides these small things of the input and output, pre-processing and other things, the main difference is that in version 2, the reference table is hosted in a public relational PostgreSQL database using pgvector. And this makes it much easier to later update it in the future versions that will come. Other differences could be that the first version only used the Euclidean distance, while the second can use both of them, and there are different parameters for that. The first version only accepted the Gene Ontology version of 2022, while the second one accepts that one, but also the latest one using a default UniProt, the ones that UniProt, the API of UniProt uses. The embeddings are also generated differently between both of them. The first version only has Proti5, while the second one includes Proti5, ESM2, and Prosti5. In the future, we may work to include more models. Also, the output is a bit different between both versions. So in the first one, it has a specific structure. In the second one, is optional. And also the output comes with much more information about what's the reference, the closest references, if you use different models, which is the closest for each model, the geo category, and many other things. Another important thing to know is that the version 1 can only be used for proteome function and annotation because of this step that I explained at the beginning, that we cannot filter the reference table. But in the second one, if you can use it for benchmarking or for proteome annotation. In this case, you would not need to filter the reference database. The first version is available as a Singularity container on GitHub, and the second one is available on GitHub as well, but can be installed locally or in an HPC cluster. It's mostly like a Python package, depending on Docker and a bit of other things. And in the documentation, it's explained how it's installed. It's like a bit like harder, like, more or less, to install it, that you need more steps, but it's more complete. Both of them accept the use of CPUs. It's recommended to use GPUs because it goes faster. But version 1 of Fantasia doesn't work in some GPUs. There are some dependency problems. That's the main reason why we decided to go to the Fantasia version 2. And the version 2, as I briefly mentioned, it has more flexibility in the choice of parameters, has many more options. So I explained it, but I explained the tool itself, but how does it work? What's its performance? So for that, we did a benchmarking in 4 model organisms, and this is the work mainly done by the Ana Rojas team, and it's already published, and then you can check it. So what we did was to take the UniProt functional annotations of C. elegans, Drosophila, mouse, and yeast, and compare these geo terms to the ones obtained using protein language models. We tested 2, Proti5 and SecPec, other deep learning methods that predict the geoterms, like DeepGo and DeepGo Plus, and Hammer, which is like a proxy or the homology-based, but it's really accurate. So what we saw is that among all the methods, Hammer, which is the one we would say based on homology, well, it's based on these Hammer profiles. It's the one that has the most precision understood in terms of getting the exact same geoterm. However, it only predicts like a few of the proteins. It doesn't cover the full proteome, that there are many that are missed. The other methods are able to predict like all of them, or virtually all of them. However, the precision of the prediction is not that high, so especially for DeepGo and DeepGo Plus, where there is a really high proportion of geoterms that are unrelated or maybe related. But in the case of the language models, T5 and SecVec, the proportion of geoterms that are the exact one is really high. In addition to that, we tested how we could, in a real differential gene expression experiment, if the geoterms predicted by Proti5 or the other methods are able to recover the original, the real result that we were getting. So we did a geo enrichment using TopGo, and then we compared the similarity between the geoterms of the Go, the real geoterms, and the ones we predicted. And you can see on the right that the SecVec and T5 have a really high semantic similarity, even if we start removing the less, the ones, some geoterms that we would say less accurate or less reliable, so sort of, but still they still are quite similar. In the case of Hammer, is lower, but because we have lower geoterms assigned, And in the case of DIGO and DIGO Plus, they were behaving quite erratically, sort of. And as we started removing, like we were getting less, and they were not able to really reproduce the results that we were getting. So more or less, like the summary of the benchmark was that the protein language models that are marked here with the Sphinx square is that they have a higher protein coverage than other methods, than other deep learning methods, in terms of that they annotate the proteins. They are much more precise, informative in terms of how specific they are and less general. They are more similar with the original ones, and also they are able to recover these functions. So, okay, in model organisms, it seems that these models work. What about in non-model organisms? In this case, we would not be able to perform a benchmark. But how many proteins are we recovering with annotations? So we tested it in this data set in MatterDB that has around 24 million genes from almost 1,000 animal species, because when we use homology-based methods, like in this case we use EgnoMapper to functionally annotate the different proteomes of this species, we got like around 50% of all the genes we remained without any annotation. And this part that we don't have annotation is what we call we call ourselves like the functional dark proteomes. So now we tested if we could, what would happen if we run Fantasia. So with Fantasia, we were able to annotate almost all of them. Okay, so Fantasia is able to annotate this. We know that it works in model organisms and it's good. Okay, what are the new functions that we are getting? So we performed geo-enrichments of the geoterms that were not annotated by homology, and wanted to have a general description of this biology that we were missing. So there were some geoterms that were common to all of the phyla, and they were mostly related to the viral response, transposable elements, immunity or toxin, which are known fast-evolving processes in animals in this case. So it's interesting this, but also we also got deeper into some specific animals. So it's in the case of tardigrades, for example, that are these animal phyla that are really small animals, are able to survive in very extreme environments, including out in space. And as I briefly mentioned before, like 40% of the genes do not have homology to the reference databases. So when we did the gene ontology enrichment of these terms that were not found based on sequence similarity, we identified some terms that were indeed related to this extreme survival that these animals have, like response to UV light, water deprivation, heat, or osmotic stress, suggesting that at least we have some putative new candidates that we can explore to better understand the biology of these tardigrades in the future. Not only that, but we also discovered some examples in model organisms. We've identified one protein that is intrinsically disordered, which means that the structure is quite flexible. The only information available in the database is what was expressed in the somatic cells of tested in the male germline of Drosophila. And when we ran Fantasia, we discovered that the geoterms predicted were involving the meiotic cell cycle. So it was quite congruent to the information that we knew of the gene expression. So we think our tool is good for this first exploratory step. That we know nothing, but now we can start exploring this unknown part. So for us, for my group that we work mostly in non-model organisms, Fantasia is like a way that we have to illuminate this hidden part, this dark part, that we knew nothing about it before. And I hope that you enjoy the talk. Thank you for your attention. And I would like to thank first the Connection for inviting me to give the talk, but also to my lab, to Rosa and Anna especially, from both the leaders of both team and the main heads behind this, for allowing the connection between both teams, the other people involved in the project, and all the funding that has helped. And the work is under review currently, but you can check the preprint in here.